Hello and welcome. Today, we are diving into one of the most incredible processes in all of biology. We're going to follow the journey of a single particle of light, a photon, from the moment it enters your eye all the way to the first whisper of a neural signal that your brain gets to interpret. It's an amazing story of physics, anatomy, and some truly mind-bending biochemistry. So let's get started on our journey from photon to perception.
So first off, just take a look at this image. What do you see? Maybe you see a face, or maybe you see a person on a horse. What's so wild is that what you perceive isn't just a perfect objective snapshot of reality. Vision is totally subjective. Your brain is constantly comparing this highly altered, processed sensory input with a lifetime's worth of knowledge it's built up about the world. And today, we're going to explore that very first crucial step of altering that input. All right, so here's our game plan. First, we're going to talk about the raw material of sight, light itself. Then, we'll take a tour of the eye's anatomy, before zooming way in to the microscopic layers of the retina. After that, we'll get into the real magic, phototransduction. And finally, we'll see how the very first layers of processing get started with bipolar and ganglion cells, which shape the information before it even thinks about reaching the brain.
Okay, let's begin right at the beginning. We have to understand the fundamentals, the physics of light itself, because that's the stimulus our entire visual system is designed to detect.
So what is light? Well, it's a form of electromagnetic radiation, and it travels through the universe as waves. Now, our eyes have evolved to be sensitive to just a tiny sliver of all that radiation. We call it the visible spectrum. And this little band ranges from about 380 nanometers, which our brain sees as violet, all the way up to around 750 nanometers, which we perceive as red.
And it is truly remarkable just how small our visible window on the world is. The full electromagnetic spectrum includes, everything from super high-energy gamma rays with their tiny wavelengths to really low-energy radio waves that can be meters long. And a key thing to remember here is that shorter wavelengths, like violet, pack a lot more energy than longer wavelengths, like red. Now, for us to see anything at all, light has to actually interact with the objects around us. And this happens in three main ways. Light can pass right through an object, that's transmission. It can bounce off an object, which is reflection, or it can get soaked up by an object, and that's absorption.
And of these three, absorption is absolutely critical for vision. It's the moment light energy is transferred to a particle. So, for example, a black shirt looks black because it absorbs all the wavelengths of visible light. A green leaf, on the other hand, absorbs all the wavelengths except for green. It reflects that green light, that light enters our eyes, and the very first step in seeing that leaf is for special pigments in our eye to absorb that reflected light energy.
The other really crucial property of light for vision is refraction. This is simply the bending of light rays that happens whenever they pass from one medium to another. You see it all the time, right? Like when you look at a straw in a glass of water and it looks like it's bent.
And this is exactly the principle our eyes use to focus the world. The cornea, which is the clear outer dome of the eye, and the lens, which sits right behind it, both use refraction to bend those incoming light rays and focus them perfectly onto the light-sensitive tissue at the very back of the eye, which is called the retina.
Okay, so now that we've got a handle on the properties of light, let's take a look at the incredible biological camera that evolved to capture it, the human eye.
Here's a look at a cross-section of the eye. Light first hits the cornea, and that does most of the heavy lifting when it comes to refraction. Then you have the iris, that's the colored part of your eye, which controls the size of the pupil to let more or less light in. The light then passes through the lens, which does the fine-tuning of the focus, and finally that light lands on the retina, the neural tissue lining the back of the eye, where our story really kicks into high gear. All the neurons there eventually bundle their axons together to form the optic nerve, which carries that visual signal off to the brain.
Now, if you were a doctor looking into someone's eye with an ophthalmoscope, this is pretty much what you'd see. This is the retina. It's actually the only place in your entire body where you can look in from the outside and see a part of the central nervous system and its blood supply directly. And we can see a few key landmarks here.
Right in the center, you have the macula, and at its very heart is a tiny pit called the fovea. The fovea is responsible for our sharpest, most detailed central vision. You can also clearly see the blood vessels that nourish the retina. And that bright spot where all the vessels seem to meet, that's the optic disc. That's the exit point for the optic nerve. And because there are 0 photoreceptors there, it creates a blind spot in your field of vision.
Okay, now let's zoom in. The retina is not just some simple sheet of light detectors. It is a deeply complex, multi-layered piece of your brain, basically.
This is where the first and arguably some of the most important processing of visual information happens.
And here's something that is absolutely fascinating, and honestly a little backwards. Look at how the retina is organized. Light comes into the eye, and it has to pass through several layers of completely transparent neurons, the ganglion cells, the bipolar cells, before it finally reaches the actual light-detecting photoreceptor cells at the very, very back of the eye.
So, the retina has 5 major types of neurons, kind of like a cast of characters. You've got the photoreceptors, the rods and cones. They're the light detectors. Then the bipolar cells, which are the connectors. They link the photoreceptors to the ganglion cells. The ganglion cells are the final output neurons. They're the ones that talk to the brain. And then you have horizontal and amacrine cells, which are these side chatter neurons that modulate signals across the retina.
And this cast of characters is organized into these really distinct layers. At the very back, you have the pigmented epithelium, which acts like a support system for the photoreceptors. Then the photoreceptor layer itself, followed by layers where all the cell bodies and their connections live, and it all ends with the ganglion cell layer, which is the closest layer to the inside of your eyeball.
Now, our photoreceptors come in 2 main flavors, rods and cones. Think of rods as your night vision specialists. They have just one photopigment, so they only see in black and white, but they are incredibly sensitive to light. Cones are the opposite. They're your daytime, full-color specialists. There are 3 different types, which gives us color vision, but they need a lot more light to work. And they're packed into the fovea to give us that super sharp, high-definition vision.
This difference between rods and cones leads to a really important trade-off in how the retina is wired up. In the fovea, where you have all those cones, there's very low convergence. Often, it's a one-to-one connection from a cone to a ganglion cell. This gives you amazing resolution, amazing detail. But out in the periphery, it's the opposite. You have high convergence. Lots and lots of photoreceptors, mostly rods, all pool their signals onto a single ganglion cell. You lose resolution, but you gain a massive amount of sensitivity. It makes it much easier to detect a faint glimmer of light in your peripheral vision.
So, we've got light finally hitting the photoreceptors. But how does that light energy actually get converted into a neural signal, a language the brain can understand? This process is called phototransduction. And honestly, this is where things get really, really wild.
Okay, so here is probably the most surprising, most counterintuitive fact about vision. In total darkness, your photoreceptors are on. They are active. They're sitting in a state called depolarization, with special sodium channels held wide open. And because of this, they are constantly releasing a neurotransmitter called glutamate. When light finally hits them, it actually causes them to hyperpolarize. They become more negative. This slams those sodium channels shut and decreases the amount of glutamate they release. So the take-home message is this: light turns photoreceptors off.
Let's just break down how this so-called dark current works. So in the dark, you've got an enzyme called guanylyl cyclase just churning out this molecule called cGMP. You can think of cGMP as a key. It binds to those sodium channels and physically holds them open. The constant flood of positive sodium ions into the cell keeps it depolarized, and that depolarization is the trigger for the constant release of glutamate.
Now, watch what happens when a single photon of light shows up. It gets absorbed by a photopigment, which causes a little molecule inside called retinol to change its shape. This shape change kicks a G protein called transducin into action. Transducin then activates an enzyme, phosphodiesterase or PDE. And PDE is basically a cGMP-destroying machine. It just starts chewing up all the cGMP. With no cGMP keys left to hold them open, the sodium channels slam shut. The flow of positive charge stops, the cell hyperpolarizes, and the release of glutamate just plummets. It is an absolutely incredible biochemical chain reaction.
So that signal from the photoreceptor, this sudden drop in glutamate, now hits the next cell in the chain, the bipolar cell. And right here, the signal gets split into 2 parallel pathways. One channel that's basically for detecting light, and another that's for detecting darkness.
And the magic trick here all comes down to the kind of glutamate receptor the bipolar cell is using. You see, off-center bipolar cells have standard run-of-the-mill AMPA receptors. For them, glutamate is an on switch. It's excitatory. So in the dark with lots of glutamate, they're on. When light hits and glutamate goes away, they turn off. But on-center bipolar cells have this weird special receptor called mGluR6. For this receptor, glutamate is an inhibitory signal. It's a stop signal. So high glutamate in the dark actually keeps them turned off. When light comes along and gets rid of that glutamate, the inhibition is removed and the on-center cell finally gets to turn on and depolarize.
So just think about that. It is so crucial.
The exact same signal from the photoreceptor, less glutamate, produces the complete opposite effect in two different cells sitting right next to each other. And it's all because they're using different receptors. This is how the retina creates separate parallel channels for processing light and dark right from the very start of the process.
But this clever split into on and off pathways, that's just the beginning. The signals now flow to the ganglion cells, which are the final output neurons of the retina. And this is where the processing gets even more sophisticated.
Every single ganglion cell has what we call a receptive field. You can think of it as its own little patch of retinal real estate that it's in charge of monitoring. A light stimulus inside this specific area is what will change that ganglion cell's firing rate, how many action potentials it's sending to the brain.
And what's really important is that these receptive fields aren't just simple spots. They have this ingenious center-surround design, like a donut. Light that shines on the center, the donut hole, has the exact opposite effect of light shining on the surrounding area, the donut itself. The center response is created by those direct connections we just talked about. But that opposing surround response is created by those side chatter neurons, the horizontal cells, that link up neighboring photoreceptors.
So let's take an off-center ganglion cell as an example. It's connected to an off-center bipolar cell. Now, since light turns its bipolar cell off, if you shine a tiny spot of light right in the center of its receptive field, the ganglion cell gets quieter. Its firing rate goes way down.
But if you take that spot of light away, creating darkness in the center, its photoreceptor and bipolar cell get more active. This excites the off-center ganglion cell, causing it to fire a huge burst of action potentials. So this cell is effectively a dark detector. It shouts to the brain, hey, it just got dark right here.
And this is just a fundamental point to remember. All those other cells in the retina, the photoreceptors, the bipolar cells, the horizontal cells, they all communicate using these subtle graded changes in voltage. Only the ganglion cells generate true all-or-nothing action potentials. Their axons are the only information highway from the eye to the brain, sending these complex, coded patterns of spikes down the optic nerve.
Just stop and think about that for a second. All of this unbelievably complex processing, splitting signals into light and dark pathways, creating these center-surround fields that are perfect for detecting contrast and edges, all of it happens inside the retina before the signal even leaves your eye. So the question to leave you with is, how does this incredible initial filtering build the foundation for everything we ultimately perceive, for our rich, detailed, and colorful visual world?